It's my pleasure to start this. And what we want to do is we want to give a talk, and I will start. Boris will do the second half of it. And what we want to do is we want to give you an idea of what this project is about, what the science of it is about, but also some of the background behind it. Because if we just tell you what the science is about, nobody will understand anything. So what we want to do is to make it accessible to everybody. So there's a bit of background and a bit more advanced as we go along. If you look at this project Excellentia, the science is divided down into 4 parts. Okay, and the first part is looking at soils and fungi, and in this case, these are ectomycorrhizas. So we want to know what influence the soils have, and I'll tell you as we go along exactly what we want to do. The second part of it is the interactions of these fungi, and in particular, the interactions with pathogens. It should become obvious towards the middle of the talk why we want to do this. Okay, so particularly we're looking at host, plant, environment, fungal symbiont pathogen interactions. So these could be fungal pathogens, these could be viruses. Okay, so that's what we want to do. The third part of it is then more towards trees, but as I'll tell you towards the end of what I'm talking about, also for fungi, is how does biodiversity relate to ecological stability? If we have more species, do we make more ecologically stable communities? That's more or less it. And the graph just shows it's one of these typical ecological stability models. Okay, I'll go into it, but that's what it is. And the fourth part is then about, well, people and forests. What do forests deliver for us in terms of ecosystem services? And what do people expect from forests towards the social part of it as the fourth part? What I want to do is I want to start, and I'm going to talk in a big block about soils and fungi, okay? And in particular, I'm going to talk about mycorrhizal fungi, okay? And because of their importance in forest systems. So why do I want to talk about mycorrhizal fungi? Because practically 80% of all genera of plants are mycorrhizal. There's almost nothing on the planet that is not mycorrhizal. They all form symbiosis between plant roots and fungi. And particularly if we look at forests, there's almost no plants in the forest which aren't mycorrhizal. Being mycorrhizal is the normal state. Okay, and what we have is, if we don't just have mycorrhizas as a name, we have many types of mycorrhizas. You can see that there's these names of these ectomycorrhizas. I'll show you what it is in a minute. We have arbuscular mycorrhizas, which I won't talk about, but probably the most widespread are arbuscular mycorrhizas. And then we have these ericoid ones, which are important in forests. And then we have orchids, which are not so important because orchids are not that common, right? Okay, but if we take these 3, ectomycorrhizas, arbuscular mycorrhizas, and ericoid mycorrhizas, we cover most of the plants across the planet and most of the plants that we find in our forests. Okay, everybody knows what an ectomycorrhiza is because many of you have eaten them for a start. Okay, these are the fruiting bodies of ectomycorrhizas, the things you find on the forest floor. And the majority of what people collect and eat tend to be ectomycorrhizas. And I just want the slide A because it's optically quite pleasant, and B, it shows diversity. It shows the diversity of the different fungi that we have. Okay, there are something like 20,000 different types of mycorrhizas across the globe. They're very, very, very highly diverse. There are many, many species of ectomycorrhizal fungi. And then that's probably a bit hard to see, to be honest, but it doesn't matter. You just got to look at the colors more than anything. What the slide shows is it shows along the bottom axis, it shows the time that mycorrhizas developed. Well, ectomycorrhizas have been on the planet for about 665 million years. That's a very long time. So most trees have been ectomycorrhizal or temperate trees for the last 165 million years in their development. And all of these fungi, all of the ectomycorrhizas, the importance of this slide is all of the ectomycorrhizas have developed from wood-degrading fungi. And these are either white rot fungi or brown rot fungi. So you can see the white rots are in this light colours and the brown rots are in brown. These fungi have been symbiotic, they're not symbiotic, free-living, and then symbiotic, and they've changed backwards and forwards. And the reason this is important is because of the evolutionary origins of ectomycorrhizal fungi, is they have retained the ability to break down organic compounds. Because wood rot, brown rot and white rot fungi break down wood and organic materials, the ectomycorrhizas have maintained this function. Okay, not as good as the white rots and the brown rots, but they can still do it. Okay, and you'll see why this is important as we go along. I can see a few people saying, what the hell is a symbiosis? What on earth was this mycorrhiza? Okay, a mycorrhiza is this symbiosis of a mutualism between a fungus, which is this one, and a plant, in which case our tree. And what do they do? Again, just so that everybody knows what we're talking about. The fungus supplies the tree with nutrients, mainly nitrogen and phosphorus. Okay, as the big ones. And the tree supplies the fungus with carbohydrates, normally in the form of sugars, normally actually in the form of sucrose, the same stuff you put in your tea. And why do they do this? Well, because being symbiotic for a fungus is a great idea, because you get this really high level supply of carbohydrates to live on. Yeah, it's the difference between eating oat flakes and chocolate, if you like, you know, probably not so healthy, but all the same in terms of energy, concentrated energy, it's the difference, right? We have this symbiosis, and this symbiosis, as you see, is very, very stable. It's a really good idea. And not only is it a good idea for trees in terms of the amount of nutrients they get, but they get an enormous number of other benefits. They can get organic nutrients, which is up the top here. They can get also mineral nutrients because the mycorrhizal fungi increase weathering of minerals in the soil. They can mediate against different stress forms. They moderate interactions with other organisms, including pathogens. And lastly, which is not really a benefit to the tree, but they're very, very important in carbon cycling. Being mycorrhizal has huge effects on ecosystems. And that's why it's been so stable, because it's really beneficial for a tree to be mycorrhizal. Okay, it's costly, but extremely beneficial. How do we know this? Let's go to the experimental. But you'd be surprised, but most of what we know about mycorrhizas are actually derived from experiments like this in the lab. This is actually one of my PhD students quite a little while back. Okay, and what we did was we developed a system where we could supply, doesn't work very well, supply nutrients into this central compartment, which just has mycorrhizal hyphae in it. And we could then look to see whether nitrogen or phosphorus are taken up. And what you see is if you add nutrients to the central compartment, then they're taken up by the fungi and given to the tree. Okay, that's very much the sort of the experimental level of how we find these things out. And we can take a slightly newer one, which isn't from my group, but another group in Germany, and they looked at phosphorus uptake. And what they could show is that phosphorus is taken up by the mycorrhizal fungi, either in the organic layer of a soil or in the mineral layer of a soil. And depending upon whether there's high or low phosphorus ability, that the organic layer at the top is more or less important. Okay, so if it's low phosphorus availability, then the mycorrhizas try very hard to extract all of the phosphorus out of the organic layer on the top of the soil. Okay, if there's a lot of phosphorus around, then they don't bother, because this is hard work. Take it up from the mineral soil. So we can see experimentally that mycorrhizas take up nitrogen and they take up phosphorus. Okay. Again, how does this work? It's sort of one of the last basics. This is a highly regulated process. What you can see is that the plant supplies this sucrose, and this sucrose is transported across to the fungus in the fungus cells, and it's highly regulated that the fungus membrane allows the movement of sugars into the cells, and in return, phosphorus, which is up the top here, is moved in the other direction. So this is not a marriage of love between the fungus and the tree. This is a very, very hard business-like relationship. Okay, you give me something, I'll give you something back. If you stop, I stop. Okay, that's the way it works. Well, that sounds like some marriages, I suppose. Yeah. So the next thing I want to talk about is, again, we have many, many fungi. Okay, we have many, many types of mycorrhizas. So this is now the mycorrhizas, not as the fruiting bodies, but on the roots. And what I want to draw your attention to is that there's all kinds of different forms. They have ones with lots of mycelium on the outside, some with very little mycelium on the outside, which are the ones up there, and some with these big rhizomorphs. So they have different shapes. Okay. And if we look across the forest, across Europe, this is for beech, what we can see is that a tree doesn't have a mycorrhiza. It has many mycorrhizas. So it picks itself a community of different mycorrhizas, different taxa. And in this case, this is what's called a rank abundance curve, but the rank is the number of fungi. So you can see that some of these trees are associated with 55, 60 different mycorrhizas. They have a whole community of mycorrhizas. We can look at this, that's for beech, and very quickly, if you look at all kinds of systems, so this is in the Austrian Alps, you find exactly the same sort of pattern. We find a lot of different mycorrhizas. And interestingly, we find some that dominate, some that are very abundant, and we find a whole load of them that are rare. They don't occur that much, but they are there, low abundance fungi. Okay, that's what this community looks like. So what controls it? Why does a tree do what it does? One of the factors that controls which mycorrhizas a tree has is simply the identity of a tree. And I'm sure all the Czech fungal collectors will know this. You get different fungi growing in different places. Well, it's because these different fungi are growing in different tree species. And what you also find is that this is for conifers and for beech, is that some are common. So there are some fungi that grow with everything. And there are some fungi that are very specific to that tree species. Okay. And this is modified by the environment. So the soil type makes a difference to which community we get. So we've now got a tree, identity, soil interaction, deciding which ones of these 65 species are going to get picked up and colonize the tree. And if we take this even a bit further, then we can see, well, what are the soil factors? Well, one is this tree host, but the driving soil factors are, one, the pH of the soil, which is an integral of many, many soil properties, and two, the availability of nitrogen. How much nitrogen is in the soil? Okay, that affects which type of mycorrhizas we get. What we've shown is what's called taxonomic diversity. It's all about species. Okay, which species do we get? But in terms of what we do, in terms of ecological function, that's not really desperately interesting. What's more interesting is how do these things work? What is the functionality of them? And what we can do is we can move from taxonomic diversity to functional diversity. And we can do this for mycorrhizas based on the amount of hyphae that's associated with them. I showed you that some have almost no hyphae, some have a lot of hyphae, and some have these big rhizomorphs, which gives a long exploration. So they look different. And again, just to remind you, if you look at my picture, you can see this one's got a lot of hyphae, this one's got almost no hyphae, and this one has rhizomorphs. Okay, so they're functionally different species, different types. And if we then look not at a community, not based on taxonomic diversity, but functional diversity, we see that for different tree species, so this is beech, and this is Norway spruce, and this is Scots pine. Now all the communities look the same. Why? Because what does a tree do? It takes a set of mycorrhizas. That one functions like that. I don't care what it's called. I just want it to work. So I have this one that functions that one. This one functions this way. I put them together. I've got all the functions I need. Right? It's a question of functionality that has to work. What does the identity is not important. So that's what they do. So suddenly now we don't have big diversity between different species of trees. We have very, very similar looking communities. And again, how do we know this? Well, most of this is known from experimental work that goes on. And this is a very famous publication. And these are my exploration types along the bottom here. I don't have the time used, but these are my exploration types along the bottom here. And what you can see was I had long distance ones and I had contact ones. And what we know is that contact ones use amino acids. They use easily available forms of nitrogen, like nitrate and ammonium. Whereas the ones with lots of hyphae tend to use these forms of nitrogen that have to be broken down by enzymes, which are these ones. So the organic matter is proteins and peptides are broken down and then taken up. It's organic, mobilized from organic matter. So we get these differences between the two. So we get a functional difference. So again, how do we know this? How do we begin to work out what a community does? That's what we're really interested in. How do these communities function? And so what we do is that all of these mycorrhizas produce these enzymes that break down organic matter. Okay? There are various enzymes. These are peptides, peptidases, and things for breaking down lignin or cellulose or organic phosphorus. We take these enzymes and we measure them on roots. And we create what's called an enzyme activity profile. So we take one little mycorrhiza and we measure all these enzymes and we try and see which enzymes does this produce and how can we then relate this to its function and how can we relate it to what happens in the community. And what we do is we do things like this. So this is an enzyme. This is glucosidase, which breaks down carbohydrates. This is the relative abundance, which we already looked at. So this is how many of the species occur. And if we measure it, multiply the activity by the relative abundance, we get what's called a community contribution. How much do the particular mycorrhizas contribute to the community? So how much of this enzyme activity does the whole lot produce? So we get a measure of their functionality. And we find some really cool things. Number one is quite a lot of the function is driven by abundance. So you may not be very active, but if there's a lot of you, then you contribute quite a lot. Okay, so we get this relationship between abundance and activity on contribution. Some of them, however, are highly active, but relatively low in abundance, which is this one here, for example, this russula here. It produces a lot of an enzyme which breaks down peptides, but it's really rare. But because it has such a high activity. It makes a really big contribution. It's really, really important. It's only a little guy that you hardly ever see, but actually in terms of the ecosystem function, it's really important. If we then look at how important single mycorrhizas can be, this is another example. This one's called Heliotales, and it produces this enzyme lactase, and it produces this lots of lactase, whether it's growing on a spruce in the Austrian Alps or it's growing on a Siberian pine in Mongolia. It doesn't matter where it's growing, it's a really, really active fungus. Okay, so who you are actually does matter quite a lot with mycorrhizas. What I want to show you now is the next bit. One of the things we're interested in, all of the trees are mycorrhizal, okay? And now we want to know, okay, what's the relationship between being mycorrhizal and keeping away pathogens? Now, you'd think that we would know a lot about this because it's really, really important, right? There are mycorrhizas everywhere and there are pathogens everywhere. And I had a choice of which two papers I used that have been produced in the last 30 years, okay? Because there's two pieces of evidence that have ever looked at mycorrhizas and pathogens that are published. And I took this one because I thought this is beautiful. It's in Western Australia, and it looks at Banksia woodlands. And these Banksias are those pretty flowers up there. They're trees, but they have these beautiful flowers. And they're non-mycorrhizal. They're one of the few things that are non-mycorrhizal, okay? And they grow in these Western Australian Banksia woodlands with two mycorrhizal trees. This is Calea thymus, which is the C, and the E is eucalyptus. And so what they did experimentally was they took these plants and they grew them with or without Phytophthora, which is a very, very common pathogenic fungi. And what they found is that the Banksias are highly sensitive to Phytophthora, whereas the ectomycorrhizal trees are not sensitive to Phytophthora. And what they propose is actually the presence of the pathogen moderates the community. If there's no pathogen, then Banksia, because it grows very, very well on these soils in Western Australia, dominates the stands. But if the pathogen's present, then you get an equilibrium between the eucalyptus and the Banksia. So now we've got a real ecological interaction. Suddenly we've got a pathogen and a set of mycorrhizas, which are controlling what the whole forest community looks like. Now that's exciting, isn't it? This is real life action of small microbes controlling what we see and what type of environment we see. And then you can think of the knock-on effects, right? Banksia woodlands that have thousands of birds in them and thousands of butterflies in them. And it's because of this contribution of the the diversity within these Banksia woodlands. So you get all this diversity growing in the different species, the rest of the diversity. And it's all down to the fact that there's a pathogen in the soil which is controlled by the mycorrhizas. What are we interested in doing with mycorrhizas? Well, the answer is we're interested in looking at different types of mycorrhizas in different types of forests. And how many mycorrhizas do we get, and how does that contribute to ecological stability?
Good afternoon. In my talk part, we'll switch a little bit above ground first. And Douglas talked a lot about this diversity and how it's important. And there are nearly no better forest than to study diverse stands in the highland of Ethiopia, where we worked a lot. In this diverse stand in Taragedam, for example, you see it. There's Lake Tana, if you know your way around Ethiopia, a little bit in the north. It's in the highlands. There are around 40 different tree species, or at least the woody species in the forest. So much higher diversity than we're used to have in our temperate Central European forest, where we're happy if you have already 8 or 6 or 10 species, and we think this is the most diverse it gets. But of course, the more species you have in your ecosystem gets also more complicated. And forestry itself, or knowledge about trees, in Ethiopia is in its childhood, I would say. So they're only establishing knowledge about species, but we try to, with our partners, to spread or get the idea on the ground that basically if we want to determine, this is true for Ethiopian forest, but also for our temperate forest, what's going to happen to our forest under future climates. We first need to understand the tree or even genotype-specific traits, the properties of my species. So how is it reacting in terms of water stress, or lack of nutrients, or any kind of pathogen attacks. And if you don't have this basic knowledge, if you don't have it established, it's very hard to predict it. And the second challenge is, of course, in this very diverse stands, and also the mixed stands we're currently creating in our shift in forestry. So how are these mixed stands reacting? Are they merely a sum of the single species, so can be calculated, or are there any kind of reactions which you cannot predict from the monocultures, basically? And of course, we need to do this for all different kind of soil types, environment, theoretically, but it's very hard. So at a certain point, we need to come exactly to what Dr. said, to create functional units which allow us to predict a little bit the future of our forest under the changing environmental conditions. And what I'm particularly interested in is always roots. I love roots. Look, Douglas maybe looks a little bit at from the perspective from the fungi up, and I always look a little bit down on the fungi from the root part. And in this example, I want to we look a little bit at the functional diversity. And this is definitely something we want to do also in Excellentia, to see how the above ground traits, this is xylem vessel diameters, how much water use, how many leaves I have, how do they actually relate to the hidden half? This term hidden half for the below ground system was probably coined about 20 years ago. And since then, really a certain development started to unravel, to undig a little bit the hidden half, but it's still the lesser known part of our ecosystems. So I'm very much interested to see, for example, in this Ethiopian system, how hydraulic connectivities, for example, in the root system and in the shoot system, if they relate. Can we even predict the hydraulic connectivity of roots from looking at the shoots? Or is it enough to even take a stem core? And for example, for this system, we were very surprised that the wood density of roots, of coarse roots, and the wood density of stems was highly correlated. But both of them were not related to the wood density of branches, which of course complicates the whole system, which means I need to independently determine these parameters for all the different organs if I cannot reliably predict them. And we thought to establish a system, for example, where we had here the deciduous angiosperms, they are broad deciduous, they drop the leaves in this extended dry spells in the Ethiopian highlands. If, for example, they have a higher branches compared to the evergreen angiosperms, which maybe need to protect themselves more from cavitation, from water loss during this dry period. But this was not the case, for example. So we don't know exactly, because we know so little about the species, it's hard to find an organizing criterion, basically. Because also the Ethiopian colleagues, they don't know exactly how broad deciduous they are, when exactly they drop the leaves, and all this information and how they are relating branches, stem, roots, they are to really predict, to really say this species is water saver, or it's more spender, has this strategy to tolerate the drought, or it's avoiding the drought, to classify. And of course, for our temperate species, we're a bit further. But what underlies all these efforts is always a trait-based approach. And then, of course, traits, we try to break down our organism in the functioning of the organism in different traits of different organs, different tissues. For example, this is an example for nitrogen uptake capacity. And you see, of course, many traits related to mycorrhiza, and which enzymes they exude, for example, to make it available, but also small detailed parameters, for example, the cortex area, the part where for the endomycorrhiza, where the fungi interacts basically with the cortex, plays a role. And this is related to how big is my stele, the water transporting part. And everything is related. This relates to the root diameter, and the root diameter is also an indicator for the lifespan. So in the end, we try to break it apart into the different sections trait of an organism to put the functional relationship between these different traits together. Of course, this is very difficult, and this is also from a huge publication with 50 colleagues. We pieced it together, and we're only at the beginning. But this is at the end. This is the goal. If you want to understand how organisms function, you need to understand the functioning or the coordinated functioning of the different parts. And to make it a little bit easier, so we don't need to travel all the time to Ethiopia or need to look in our forest stands for different mixture combinations. Douglas already started, I don't know, when you established your tree diversity experiment in Bangor in early years. And in 2003, so 10 years earlier, we established, for example, a seedling diversity experiment at the Boku. And we joined this largest network of tree seedling mixture experiments called TreeDivNet. Among all these partners, we try to pool the data to get from, you see where everywhere these temperate ecosystems, here it's a little more spread. Here in Europe, we are quite good covered, but also not the alpine, not the riparian forest, and so on. There are many gaps in these data sets. To pool it together to get an idea, what are the effects of mixtures on the stability of the forest, on the productivity of the forest, on the habitat function, on the health function, all the aspects. In our experiment, 11,000 trees were easily planted and replanted the next year. Basically, we established a gradient between monocultures of Acer, Tevia, Quercus, and Capinus in this case, which are a little bit, have a little bit an ecological gradient for more acquisitive species like the fast-growing Acer plant to the more conservative resource-optimized species like Quercus. And we had this highly diverse 4-species stands, 2-species stands. We could only realize, unfortunately, for Quercus and Capinus and Acer and Tevia, just because we had only 1.2 hectare and could not realize everything in replication. And we were surprised that relatively fast this diversity effect kicked in, because many colleagues from the other experimenters said, no, it took 15 years or longer time periods before they could see any effect. For us, it was relatively fast. They planted 2013, replanted 2014, to fill the gaps again. And this is the time axis we measured every 3 years, the height, for example, or the basal area of the trees and the mortality. And you see already for Acer, for example, in 2019, so 6 years after planting, we had already a different basal area in the monocultures, in the 2-species mixtures, in the 4-species mixtures. And you see here that the significantly lowest amount is in the monocultures, basically, and much higher basal areas in the mixtures. Because here all the monocultures are also shown together, and you see there's not much difference. If they grow basically in the monocultures, the 4 species behaved relatively the same in terms of growth. But the moment you put them with different neighbors, and this is of course well known because it's a common forestry practice, we grow them together so they get a nice long stem or grow proper, and later we thin this again, we have this effect. But relatively surprising was that it was relatively fast. Around crown closure was around 2019, 2020. That depended a little bit on our different blocks, because also in a small field, not everything is so homogeneous, but relatively fast. What was the effect? This is the biomass in 2021. We did a big campaign. We see again, of course, Acer had the highest leaf biomass, relative high wood biomass, and fine wood biomass. But what was, and this is, you see here, always separated in the colors, basically in the 2-species mixtures, was the relative contribution of the different species to this component. What is probably more interesting is on the right-hand side, because you see always observed. The observed biomass value is in the dark gray, and the expected value, which is basically the average of the monoculture, so the calculated value, is in the light gray. And you see, for example, for the fine wood biomass, a significantly higher amount of fine wood biomass we find compared to the average what we would expect in the 4-species stand. The same for the stems, for the woody biomass, and also the same for leaves. So what we get in our 4-species mixtures, for example, is more leaves, more stems, more roots, if grown together. Yeah, and of course, the system, as we see in this artificial combination, is of course largely driven by ASA, because ASA contributing everywhere the major share. So the species identity matters, but of course the combination, what are the competitors, they matter also in the different systems. So what's driving this? What's driving this advance in growth? What is believed, or this is the common understanding in literature, is the enhanced structural complexity. So the better use of space and resources in my mixed stand. There are many values. Yeah, we cannot have the time to look at all of them in detail, but we looked, like I said, from the ground shape to the leaf amount to the root distribution. What we see, for example, in the mono acer stands, in the two species mixtures, they get more leaf biomass, more leaf area, but also a larger projection area, larger basal area, fine root biomass. Yeah, but this is also very species specific. Yeah, for example, Papyrus on the other hand didn't show any of these similar adaptations to the mixtures, but only smaller, the median height of the crown diameter, basically, all the crowns were low. Yeah, the median diameter of the crown shifted lower, basically a two-storage forest started to develop in this stance. Yeah, after 6 years, I think it's for 9 years, then it's quite impressive that you see already this structural diversification. And we are one of the few groups. We also looked at the root overlap, because there's always the question then in this mixed stands, do the roots segregate or do they overlap? Yeah, and we can only confirm that they're overlapping a lot. We sampled always between 3 trees. This is why here on the top you always see the Acer tilia, for example, token, and then the amount of share, how many fine roots, which part of the fine root biomass was given by which species. And you see that Acer, for example, either is there 2 Acer trees or 1 Acer tree and 2 tilias. Acer seems to be in this combination a relatively good competitor below groundhouse. They are not only above ground, as we know also from our observation in the field, where we know it's going good, at least in the beginning, first 30, 40 years, yeah, but also below ground, it's dominating this stand. Yeah, but also, yeah, Quercus, for example, Quercus is here in orange, for example, is a terrible competitor. Yeah, also not very surprising, because we know already from many experiments in Germany that, for example, Fagus and Quercus, Fagus is always outcompeting Quercus in the way that Quercus is looping all the organic or the rooting space and organic layers on the topsoil to Fagus. So obviously the strategy of Quercus to be relatively resource efficient and to save the resources doesn't really pay off when it comes about getting limited resources in a shared space. Yeah, because suddenly Quercus doesn't have access to these resources. This is definitely something we want to, in this Excellentia Center, to have this look a little bit how above ground structural diversity and below ground structural diversity, of course, complicated by all the different mycorrhizal types, the different functions, are shaping then this function of this species. And this, of course, we want to do also in the university forest, which is a unique setting to find different diverse stands. So this is just in the process identifying this, but already, this is my first scientific work. When I did my master thesis in plant ecology, I studied the competitive ability of Capinus, Fagus, Quercus, and Tilia in the driest spot in Germany, also only about 550 millimeters of rain. They are already turned out, of course, we know Fagus is a very strong competitor. Quercus is a very weak below ground competitor. This means when they're growing together, Quercus would stop to grow, and Fagus would dominate a small rooting space. We don't know yet the mechanism. How they recognize. Is it only indirectly by the depletion of nutrients, or are there direct signals like volatiles, hyphal networks, what we hear so much about in the media? What are the mechanisms, how they recognize that there is not a conspecific competitor, not from the same species, but a different competitor basically in their direct neighborhood? There's barely nothing known about this. But now we talked a lot about biomass, and in general, it is about a lot of different root traits. We have morphology, we have the anatomical traits, we have these mycorrhizal symbionts. And this graphic we try to symbolize a little bit where are already connections in the literature between all these different aspects of root functioning, for example. Yeah, it's also relatively thick lines demonstrate there's a good connection, of course, logically between root morphology and root architecture, because if my root is thin or thick, makes a difference on where I can put my roots and how I can develop the architecture basically of my system. But other combinations, for example, mycorrhizal association, how this is really related to the architecture of my root system, we could not find much evidence. Yeah? And of course, in the next years, we hope to contribute to filling some of these missing links. Again, yeah, once we have these missing links, we will be not only able to look at mass distribution under different competition regimes, but look at more specific traits. Yeah, in this pot experiment with my student from Iraq, he studied Acer and Fagus in action. Here's always the monoculture, the intraspecific competition and the interspecific competition. We see that also traits like root respiration. Yeah, so how much carbon is consumed by my root system, which is actually not good because I cannot produce fruits from it or seeds from it. I cannot grow above ground. I cannot outcompete them. Yeah, this is significantly reduced, likely by a reduction in the fine root nitrogen concentrations. On the other hand, on this effects, potential activity of these different enzymes or selection of the enzymes Douglas mentioned earlier, we could not find very large differences. Here on this PG, for example, we found in this interspecific competition a reduction, but we did not check in this study because Douglas was not involved if this is related to a change in the ectomycorrhizal community. Yeah, but in between, the Acer has mostly endomycorrhizas, so it's a bit complicated in this case, or related to different activities. The whole root science, and this is this keeps me up at night, basically, my colleagues of this very small root ecology, we cannot even really agree how to classify it. And this is hampering the whole field a little bit because in the literature we find this description of roots, for example, either short roots or longer root tips, mycorrhized, non-mycorrhized, fine, or very often the most common description is the diameter-based description. We have the fine roots under 2 millimeters and we have the coarse roots over 2. But if you look, for example, at these 3 different species, and you look at the scales, what is actually the functional unit? Cannot be related to diameter. It must be related somehow to the branching pattern, maybe to the age. So we're trying to find ways to classify a root system to compare apple with apples and pear with pears, and not basically very thin roots with very thin roots, which can mean something totally different in different species. Just why root system is why root ecology is maybe not so advanced like leaves, because a little bit more complicated to classify. So this was a lot about roots. So we want to, in the last part of this presentation, want to a little bit talk about the overall society functioning of our forest systems, beside the obviously carbon sequestration and provision of wood or non-wood products, so society needs. And there I had Naomi Frotier, she did a master, or she's still doing a master studies with me. She wanted to look at how are these different tree diversity levels in our bee tree experiment influencing the insect diversity. And there is a worldwide initiative where they look at tree holes. You see in old core forests sometimes water-filled tree holes. Because our stands are so small, we mimic them and go into IKEA and buy hundreds of these beakers and fill in some water, take them from the outside, and then we put some litter inside and incubate them over one season. So these aquatic microcosms are our fake tree holes. And after 3 months of incubation, we followed the water level. We basically harvested them and looked which insect species basically are inside. And the overall result is that the tree diversity destabilizes these aquatic tree hole habitats because, first of all, we see the water level over time. T78 is about August. The August in eastern Austria are very hot. So many of these in our outside control all the microorganisms were not in the forest, then they were dried out. Anyhow, this is the orange line. We had a little bit water in the monocultures in the 2-species stand, and the highest water level we had in the 4-species mixtures. This, of course, is beneficial if I'm not allowed to dry out to stabilize my insect community. And indeed, the numbers of these aquatic insects in the 4-species mixtures, she quoted here all, they are significantly higher compared to the monocultures. The mixtures and also the diversity, relative to the highest diversity we found, was significantly higher in the mixtures. So from the habitat provision, we can say different species in combination, of course, provide different niches for different aquatic species here, but also the different litter qualities which were inside, the stabilization of the habitat itself, that the water stays inside to provide more insects and a higher diversity of insects provide them more. The second aspect, which is important, particularly if I look out of the window for urban forest, but not only, is air quality. Particulate matter, particularly the ones under 2.5 micrometers in size, are extremely cancerogenic, basically. We inhale them. There are a lot of studies related to heart diseases and many cultural sicknesses, basically, from this fog in our forest. But nevertheless, there are relatively, until recently, until 2010, 2011, relatively few studies, surprisingly, which look at how the green infrastructures of our urban environments or forests are helping us to clean the air from these small particles. It's surprising. Very few studies before, then suddenly it starts. But also, not surprisingly, in countries which have very big problems with the particulate matter concentration in the air. This is, for example, China and Poland. And then there's a very active group in Italy, but then not so many. And Czech Republic is also not the big countries like Germany for publications until 1994. It's really not much, and a very important factor. So what are the traits of species which shape their potential to accumulate or to clean the air and dissolve, absorb this particular matter? Of course, you can differentiate them on the tree level. It's going to be the crown geometry, the tree height, the tree age. But on the leaf macrostructure, we have leaf shape, leaf sizes, and on the microstructure, very important, the numbers of hairs, trichomas, or the appearance of a wax layer, where the particulate matter can be stuck. So we did recently, this year, published a meta-analysis from all these papers, and this is also was known but not summarized, basically, where we checked in different polluted environments which kind of trees are more effective in removing this particulate matter. So we had here the conifers and broadleaf. The conifers are in dark green, and we know all the needles that are very waxy. So, and they stay along also the whole year. So particularly heating period, for example, in our temperate systems, they're very important to clean our urban air with conifers, because the waxy needles absorb all the particulate matter. And this was true for all along the gradient. In the medium, also this information, the meta information in these articles was very weak, so it was very hard to classify really this is a highly polluted, low or medium polluted environment. And then one of Douglas' PhD students before already, he went to Bee Tree again and looked at our maple, lime, oak, and hornbeam. How are they doing in terms of their absorbance capacity? So we have this PM 2.5 here on the bottom, and PM10, a little bit bigger particle size on the top. And you see there's a clear difference between Acer and Capinus trees, where the Capinus trees, the hornbeam, for example, for the PM10, binds about more than 2 times more particulate matter of this size class than the Acer trees, not in the maple trees. So there's a clear species-specific ability based on the traits, like the wax layer, the leaf size, and so on. But of course, the interesting question is now, how does the mixture affect this? Now we still look at individual tree species. And there he calculated observed and expected values for our 2-species mixtures, Acer with Tilia and Quercus with Capinus, and the 4-species mixture all. And in in the ASA and the TVR, we see again our observed values were higher than the expected values from the monocultures. Yeah, and highly significant in our 4 species mixtures. So how does it come? Did they change? One important factor, of course, was the increase in leaf area. Yeah, because our mixtures have overall a higher leaf area. They have a better cleaning function for this particular matter. Yeah, because this is the overall particulate matter accumulation basically per ground area, because they are just they have this higher leaf area. So mixed forests are better for cleaning air. This is the take-home message for this part, because they can provide a larger surface area. It was a quick overview on things we're interested in, and we hope to realize in Excellentia. We're building a team. And now next to our office in the 2nd floor, there is also a list of potential master and science, master and bachelor's thesis, for example, we can offer right now, from the ectomycorrhizas of oak to the nitrogen usage of oak. But we're also interested how grafting of forest trees influence root traits. So how are these above ground and below ground traits coordinated the particulate matter accumulation? Be of interest to us would be cooperation with BOKU. We have the equipment still. Yeah, tree diversity, productivity of riparian forest, soil compaction, also management effects are of interest for us. So how management affects basically ecosystem functions, or through influencing the rooting space, so to say. Rooting depth of mixed species stands. Is this significantly driven by the mixture, or is it really genetically preserved? How deep they grow, and so on. And we're also happy, of course, always if you have good ideas, you say this is really something which is important in my region. This is important. I'm interested in this, to discuss it with you and see these are possibilities we can realize. Now, if we want to do good science, we're happy to cooperate with everybody who wants. So thank you very much for your attention.